Hi everyone, this is Mr. Sinti, and today we're going to be talking about proteins. Proteins are another one of those macromolecules that are most important in biology. Along with carbohydrates, lipids, proteins are there as well in terms of importance. The name itself actually means of first importance. But what I want to say about proteins is that they're really instrumental, as it says here, in almost everything that the organism does. Everything in the cell involves proteins, and one of the things about proteins right out of the gate that I want to mention is that uh, proteins are polymers. And the fact that they're polymers means that they're made up of repeating subunits of monomers. So like a chain of pearls like this. And so each one of the pearls is made up of this thing, which is the monomer, which is an amino acid. And so as you can see from this next picture right here, if I were to erase that, um, amino acids are a little bit different than monosaccharides in the fact that they're different. Each one of them is different from the other one, and there's 20 different kinds. There's 20 different kinds of monomers. Amino acids are also called peptides, and so if you understand that, one of them is a peptide, two of them is a dipeptide, many of them are polypeptides. And so we could simply say that a protein is a polypeptide. So poly meaning many of these peptides held together in a long chain. So a polypeptide is a protein. And so when you look at the specifics of an amino acid, what you could see here is, use my little highlighter, this region right over here is considered to be, whoops, didn't push the button. This little area right here is considered to be the amino part, right here. And then the acid part is considered to be, let's do this, carboxylic acid is considered to be this part right here. And so what you have is an amino acid, and how they differ is in these particular R groups. So in other words, there's 20 different things that are coming off. And you're like, okay, 20 different things coming off the central carbon. What does that look like? If I showed you this picture, go to the next slide, you can see that the R group is a CH3 coming off the bottom of the molecule. And like any functional group, the R group will determine the particular properties of the amino acid, whether it's polar, whether it's nonpolar, whether it's electrically charged. And so here are some examples. It looks kind of complicated, but don't worry. This highlighted area is the R group. So for glycine, which is one of the 20 amino acids, it's hydrogen. Alanine, it's CH3. Valine, it's right here. But what all of these have in common is that all of these are nonpolar. Translation, they're hydrophobic R groups. So water doesn't want to mingle with this. So it's kind of interesting. If you were to consider the next group of amino acids, like for example, these ones are all polar. As you can see, OH, OH, SH, OH. So water would stick to these. And the significance of whether or not it's polar or nonpolar, or these happen to be negatively charged, these are positively charged, these considered to be basic, acidic, is that collectively, when you put the peptides together in a chain, what you end up getting is these R groups sticking out. Let me see if I can highlight that. Let's pick blue. So this is the R group that's sticking out from each of these amino acids. So if I have one amino acid connected to another one, this is the R group that's coming out that's highlighted. R group, R group. And if I wanted to add one more, you might recall this is known as a dehydration synthesis. So water is released and then a new peptide is added. And so as over here you could see there's now 3 peptides. And so these particular R groups will give the protein its uniqueness. And what we mean by that is this. When you consider this amino acid, this one, and this one, it's what happens in the body is when we eat a protein, like from food, and it comes in, what's going to happen is that water is going to come in our digestive system, and it's going to cleave this through hydrolysis. And so we've got these now a pool of amino acids that are present in the cell. And then the cell links them together like this. There's 4 of them. It'll link them together to make a chain. And when you make a chain like that, you're starting to see how it can increase. And let me show you. Like, here's 3 of them held together. All of them have the same R group. And what happens is, this is an interesting picture. It's like, how do we go from that other picture to this one? This one is a computer X-ray crystallography enhanced image of what a protein looks like. What's fascinating about it is when you put all these amino acids together and you fold them up, they form a 3-dimensional structure. And what's interesting about that is because of all the R groups are positive and negative, it's sort of like magnets. They stick together and they form these interesting structures like this. And this is a very important idea right here. What it looks like, what the protein looks like 3-dimensionally, will give it its function. So in other words, very simply stated, if a protein, sort of like a key, if a protein has a particular shape, it's going to work or not work depending on what it's trying to do. Let's take a look over here. A couple of examples of that. Here's a bunch, not showing the detail, but here's a bunch of amino acids all linked together. Notice it sort of looks like a necklace or a long chain. This is what we mean by a polymer. But unlike starch or cellulose or glycogen, a protein is made up of different amino acids. And what's interesting, there's a lot of diversity that can arise from this. Because if you think about it, if a protein is thousands of amino acids long, the sequence, and if there's 20 different amino acids, the sequence is almost infinite in terms of all the possibilities that you can have. And what the result of that is that it forms an interesting 3-dimensional shape because the R groups are interacting. And so one of the things that we want to talk about that I'm going to mention in the next video is enzymes. A protein can act as an enzyme, which is something that speeds up a chemical reaction. Because of this 3-dimensional shape, it gives a lot of versatility to the molecule. Let's see, let's go over here. Take a look here. So, not all proteins are enzymes. Some can be antibodies. I'm not sure if you've heard this before, but antibodies are really important. They keep us alive. Our white blood cells in our body fight off germs. And one of the ways in which they fight off germs is they simply eat the germs. But there's a special type of white blood cell that makes a protein called an antibody, and it really looks a little bit like this diagram here. It looks like the letter Y. And it's like, how can it look like the letter Y? All of these are amino acids linked together to form this unique shape. And these little Y-shaped proteins circulate in the blood, and they attach to foreign substances. Like, for example, if you had some sort of bad bacteria, these are like bullets that go around and attach to bacteria and destroy them. So they're rather important. So proteins can defend our body, they could speed up chemical reactions, they're found in the cell membrane, they do all sorts of things. And all of their functionality, all of their diverse functionality are clearly the result of their diverse shape, because the chain of amino acids folds into what looks like a pleated sheet or a pleated skirt. Some of them can fold into helices, and then collectively, if you put this all together 3-dimensionally, you get this kind of an elaborate shaped molecule. And then what's also clever is that if you had more than one polypeptide chain, you can actually attach those together and get something really large. An example of that would be if you've heard of the protein hemoglobin. Hemoglobin is made up of 4 separate polypeptide chains, and that's found inside of red blood cells, and it holds on to oxygen. It's really important. And so that concludes our discussion of proteins, and I will continue our conversation with enzymes in the next video. Thanks for your attention, and let's see if we can have trouble turning this thing off. Let's see if we can do this. Let's do that, and this concludes. Thanks.